All right, this is the lecture for Lab 12, Respiratory System. First off, on page 125, you can cross out a few things. If you go to larynx model, you can cross out glottis. That's just the space the air travels through. For the lung model, you can cross out carina. And then down at the bottom, you can cross out pleura, both parietal and visceral, as well as pleural cavity. Those things cannot be seen on our models. All right, respiratory system is composed of the upper respiratory tract, which is the nose, nasal cavity, and pharynx, and the lower respiratory tract, which is the larynx, the trachea, the right and left lungs, and the various bronchi that branch out within each lung. Functions of the nose includes obviously being a way for air to come in, and when the air is brought in, inhaled, it is warmed, it is cleansed, and humidified, made wet, made warmer, and particulates collected by the mucus in the nasal cavity. The nose also detects odors, so we can smell, olfaction, and it acts a little bit as a resonating chamber to amplify our voice. Within the nose, we have these three bumps that protrude on the lateral walls of the nasal cavity, so it's three pairs of bumps, and they are covered in mucous membrane. And what they do is they sort of cause turbulence in the air as it goes into the nasal cavity, thereby mixing the air more thoroughly and helping to get it humidified, warmed, and cleansed. We also have sinuses that are connected to the nasal cavity. We have the frontal sinus in the frontal bone, the sphenoidal sinus in the sphenoidal bone, ethmoidal sinuses in the ethmoid bone, and the maxillary sinuses in the maxilla. These open spaces are lined with mucous membranes, filled with air, and they connect directly to the nasal cavity. All right, this brings us to the pharynx. The pharynx might be what you think of as the back of the throat. The pharynx starts off with its superior compartment. That's called the nasopharynx. So directly after the nasal cavity is the nasopharynx. Within that space, we find the pharyngeal tonsil that is directly across from the nose, where air will hit and potentially pathogens will try to break into the body. And we also have the opening for the auditory tube that goes to the middle ear. The next chamber is the oropharynx. The oropharynx is directly posterior behind the oral cavity, and it possesses the palatine tonsils. And then below that, the final part of the pharynx is the laryngopharynx. The laryngopharynx is directly superior to the larynx and esophagus. Here is our lovely larynx model. So this bone here, that is the hyoid bone. This flap up here is the epiglottis. Here is the thyroid cartilage, also known as the Adam's apple, that sticks out anteriorly. And the very end of the larynx is the cricoid cartilage. So from hyoid bone down to cricoid cartilage. The epiglottis is an extremely important structure because when we swallow, it folds down and covers the opening to the larynx, thereby preventing food and drink from going into the larynx. Here's another view of the epiglottis. Again, it'll cover the space, the glottis, when we swallow. Here is a mid-sagittal section of the larynx model. We can see there's these two folds. The superior fold is the false vocal cord. The false vocal cord closes the glottis when swallowing. And below that, inferior to that, is the true vocal cord. The true vocal cord is the cord that produces sound. So we have a pair of false vocal cords and a pair of true vocal cords. There are also lots of little muscles involved. There are tiny intrinsic muscles within the larynx that can tighten or loosen the true vocal cords in order to increase or decrease the pitch of the sound we produce. And there's a lot of extrinsic muscles attached to the larynx that help to elevate it during swallowing. Inferior to the larynx is this long tube. This long tube is called the trachea. The trachea will carry air down to the lungs. And as you can see, the trachea has many, many cartilaginous rings that help to form its structure and keep it open. The end of the trachea, it splits into the right and left main bronchi. The right and left main bronchi will enter the lungs. So they're the first structures carrying air to enter the lungs proper. gives us our two lungs, the right lung and the left lung. The right lung has three lobes, the superior lobe, the middle lobe, and the inferior lobe. The left lung has two lobes, the superior lobe and the inferior lobe. There's also this little space here called the cardiac notch found in the left lung. The cardiac notch is basically a space that's there to make room for the heart, because as we know, the heart is a little larger on its left side than on its right side. In this picture, we also can see the diaphragm. The diaphragm is the critical sheet-like muscle that is necessary for breathing. Without it, we would not be able to breathe. All right, the main bronchi enter the lungs, and then they split into the lobar bronchi. The lobar bronchi, smaller tubes with plates of cartilage holding it open, and each of those lobar bronchi go to one of the lobes of the lung, so there should be 3 for the right lung, 2 for the left lung. The lobar bronchi then branch into the smaller bronchi, the segmental bronchi. The segmental bronchi, also held open by plates of cartilage, go to the different segments, different specific regions of the lungs. The bronchi then branch into smaller and smaller bronchi until finally they become what are called bronchioles. Bronchioles have no cartilage, so they're just held open by smooth muscle tissue, and eventually the terminal bronchioles. These are all the structures that are carrying air deeper and deeper into the lungs. So again, trachea to main bronchi to lobar bronchi to segmental bronchi, and then those continue to get smaller and smaller, eventually becoming bronchioles, and then the terminal bronchioles. And the jobs of these structures is to just carry air deep into the lungs. The bronchiole eventually lead to alveolar sacs, and within the alveolar sacs are these little compartments called alveoli. Alveoli are very, very thin, and this is where the gas exchange between the air in the lungs and the bloodstream occur. So in this slide here, these very thin little lines here, these are representing the exterior of the alveoli. So you can see the alveoli are super, super thin, mostly empty space where air will be found. So again, terminal bronchiole branches into the alveolar sacs. So these grape clusters are the alveolar sacs. Each individual grape is an alveolus. The alveolar sacs are covered in pulmonary capillaries so that blood can be brought to the alveoli. Take a close-up look. Here is a pulmonary capillary. Here is the alveolus. We look at where they're connected. We see it's just two thin cell layers thick. So all it is, this respiratory membrane, is the squamous alveolar cell, the squamous endothelial cell, and then the shared basement membrane. Between these two thin layers, we'll have oxygen going from high concentration in the air in the alveolus to low concentration in the bloodstream, and carbon dioxide going from high concentration in the bloodstream to low concentration in the air in the alveolus. So this is where the gas exchange occurs. Now, if you go have breathing issues, you may go see a specialist, and they might have you use a spirometer. A spirometer just measures how well you're able to breathe out, how much air you can breathe in and out. It can be very complicated, very simple. We would have a really simple one that we used in the lab, but unfortunately, we didn't get to do that. And they're called, again, spirometers, measuring ventilation, how much air we can breathe in and out. So there are different degrees of effort for breathing in and out that then move different volumes of air into and out of the lungs. So tidal volume is the volume of air in one quiet breath. So when you're at rest, breathe in, breathe out, that's tidal volume. So it is just a small amount, maybe 500, 800 milliliters. Then we have inspiratory reserve volume. This is air in excess of tidal volume that can be inhaled if you try to breathe in as hard as you can. So as you can see, you have extra space in the lungs. You can hold liters of air on top of the regular tidal volume. Then we have expiratory reserve volume. This is the excess air that's in the lungs even after you breathe in and out normally. So this, with extra effort, you could breathe this rest of this air out. So as we can see, do your tidal volume breathing, normal relaxed breathing, but then you can get rid of even more air. So that's the expiratory reserve volume. Now, the residual volume is the air remaining in the lungs after maximum expiration. So the air left in the lungs after you breathe out as hard as you can, because there's always some air in the lungs, otherwise they would collapse and that would be bad. So this area here is the residual volume, the amount of air, about a liter or so, that's always in the lungs. And then vital capacity. Vital capacity is the total amount of air that you can breathe out after breathing in as much as you can. So you breathe in as deep as you can, and then you breathe out as hard as you can. That amount, that volume, is the vital capacity of your lungs. So again, tidal volume, what you normally breathe in and out when you're relaxed. Inspiratory reserve volume, the extra space that you could fill in with air if you breathed in as hard as you can. Expiratory reserve volume, the amount of air that's in the lungs that you could breathe out if you breathe out as hard as you can. And then total vital capacity, the total amount of air that you can breathe out after breathing in as deep as you can. Boom, boom. This is liters of air. I found that I can breathe in and out about 4 liters, which is a lot actually. All right, the brain obviously helps to control breathing. We cannot breathe unless the brain, the structures in the brainstem, control and allow us to breathe. And the parts of the brainstem that control our breathing is the medulla oblongata and the pons. So they are responsible for causing us to breathe and to breathe correctly. Of course, we can voluntarily, intentionally change our breathing rate, change how deep or shallow we breathe. So the motor cortex of the cerebrum also can play a role, but that would be active, us consciously trying to do that. Blood chemistry. Turns out breathing and the rate you breathe affects the chemistry of the blood. It affects the pH, the amount of carbon dioxide, and the amount of oxygen in the blood. So we have receptors, peripheral chemoreceptors, that are in our large arteries, and they detect the amount of oxygen, the pH, and the carbon dioxide levels of the blood. There's also some chemoreceptors, central chemoreceptors in the near the medulla oblongata that detect the pH, the amount of hydrogen ions in the cerebral spinal fluid, which of course, as we know, bathes the brain. So we do not want the pH to get too off in the cerebral spinal fluid, because if it does, it could damage the brain. So our cells are always producing carbon dioxide. That is a waste product from cells producing energy. Too much carbon dioxide in the blood lowers the blood's pH. There's too many hydrogen ions, the blood pH becomes acidic, goes down. This could happen, say, when you're exercising. High carbon dioxide in the blood means you have high carbon dioxide in the cerebral spinal fluid, which means the pH is getting acidic in that as well. This is the stimulus for breathing. We breathe in deeper and faster when we exercise to get rid of carbon dioxide. It is not oxygen we're worried about, it is getting rid of excess carbon dioxide. If you hyperventilate, if you breathe in and out rapidly, super fast, you will raise the level of carbon dioxide, decrease your pH, make it more acidic. And if this is uncontrolled, you will go unconscious, because the brain will say, this is dangerous, we got to get you to stop breathing so fast, and the only thing it knows to do is knock you out. So the amount of carbon dioxide in the blood making the pH acidic is a big problem. Now we also have the flip side. If there's too little carbon dioxide, then there's not enough hydrogen ions and the blood pH goes too high, which then affects the cerebral spinal fluid, and this will then cause you to breathe slower, breathe shallower, to lose less carbon dioxide, thereby to keep the pH lower. So basically, you can make it easier to hold your breath longer by hyperventilating a little bit before you hold your breath. So you have your normal level of carbon dioxide in your blood. When you when you hold your breath, you can hold it for a certain amount of time before the CO2 levels get so high that you feel like you absolutely have to breathe. If you hyperventilate, you lower the carbon dioxide levels in your blood. It's now less. And so that means there's more time it takes for the carbon dioxide levels to build back up before you feel you absolutely have to breathe. So boom, look at that. You can hold your breath longer if you hyperventilate a little bit beforehand. And that is it for this lecture.